So microbes grow through a pretty unique way. They do this thing called binary fission, which means that one bacteria will sort of split to become two, and it's asexual. So what ends up happening is you have a bacteria, it copies its genetic material, sort of creates that dividing line, and eventually just splits into two. And these two are both going to be exactly the same clones of each other. The ways that these organisms can go ahead and reproduce other than that, with anything in microbe, is going to be budding. Budding is an asexual process that yeast can do, where what you'll see is one large yeast cell will sort of spurt off into a couple, and they'll just start producing these new offspring. Sexual reproduction, that's going to be in eukaryotes, so fungus and protozoan, where they find an opposite sexed organism, and they mate with it to create a unique organism. Fragmentation, which is in mold, where they'll break apart and to come into a bunch of different pieces. I wonder if you'll see this on a future exam. You can go ahead and fill this in and see if you get it right. Let's go ahead and do it together. So with binary fission, that is going to be, what is it briefly? That's going to be where an equal division of a single cell with that host. So that's going to be one cell becoming two. They're both going to have that same genetic information. Is it sexual or asexual? It's going to be asexual. And which microbes do this? This is going to be bacteria. Budding is going to be a new organism is produced from the parent organism. It is asexual, and what microbes do this is yeast. Sexual reproduction is going to be where two organisms come together and they produce a unique organism. It's sexual, and which ones do this? Eukaryotes, but the specific ones that we talked about are going to be fungus and protozoan. The last one is going to be fragmentation, and that is where an organism breaks into pieces, and each one of those pieces grow into a new cell. So the example would be mold, and it's asexual. So bacteria grow in these phases. They have a lag phase, a log phase, a stationary, and then a death phase. And if we look, the lag phase is a period of adjustment to the environment. It's going to be where it's sort of inoculated on something, let's say a host, and it's going to sort of take over. Log phase is where there's going to be a rapid growth that makes bacteria more permeable. This is where they're the weakest, but this is where they're reproducing the most. So this would have been the log phase, the lag phase. Sorry, this is the lag phase. This is the log phase. Can you see how quickly they're growing? And that's when they're most susceptible to chemicals and things like that, so medication. So we're always trying to get them into that log phase and then treat them. Stationary phase is where they're going to have equal amounts alive and dead. They're the least resistant to chemicals at this stage. And then the death stage is where they've produced so many chemicals that maybe used up all of the sugars and waters that have started to change the pH, and they start to die. So the ability to cause a disease is called a pathogen. Pathogen is organisms that's capable of causing a disease. Patho means damage, gen means making. Pathogenicity is the study of disease, and pathogens are just the disease developing. So pathogenesis. And non-pathogenic are organisms that are incapable of causing diseases. There are these things called opportunistic pathogens, which means that under certain conditions, these organisms are really good for us, but in the wrong spot, they're really bad for us. So for instance, if we go ahead and look at E. coli, E. coli in our large intestines is great. Without it, we'd have major problems. But E. coli in our urinary tract is not good, and that is going to be UTIs, or the other thing is E. coli in our eye, that's going to be red eye. So one location, awesome. If it has the opportunity to go somewhere else, it's going to be bad for us. Sources of disease. We have our normal flora, so they are on us, they're there to protect us. They're there in one location, and they usually try to stay in that one location for them because it's good for them, but they could be all over us. So for instance, this is going to be some in our stomach lining, in our small intestines. This is going to be H. pylori. We can also, Cisterobacter is just named for H. pylori. You can see these orange ones. These are going to be Staphylococcus or S. epidermis. These are going to be in our respiratory tract, nasal tract. And then this is our large intestines. This is going to be E. coli. That's these stringy guys. These round ones are going to be lactobacilli. These are those probiotics that you eat, those pro-antibiotics, and they help this area. I'm never going to ask you about these names. I'm only interested in that they are around you and they do help you. We have these different types of relationships with these bacteria. Symbiosis. These all fall under the relationship of symbiosis. So two different organisms living closely associated. Commensalism is where one is benefiting and the other one isn't really being harmed or affected. So there's lots of things growing on our skin, and they don't really affect us really badly, but they are feeding off of our dead skin, so they like being there, but we don't really care. Mutualism is where both benefits, so this could be like E. coli in our large intestines. E. coli benefits because it's getting sugar and a warm environment and all the ability to grow. We're benefiting because E. coli produces vitamins for us and shares them with us. A parasite could be something that is doing damage to us. So one is benefiting and the other one isn't, and it could be E. coli again. So E. coli is really happy to be in your bladder. It's getting liquid environment, sugars, all the things it needs, and for us it's hurting us, so it's bad. So same organism, two different spots. One, it's really helpful to the point that if you didn't have it, you'd die. One, it's just a really bad annoyance, and if you don't get rid of it, it goes up to your kidneys. You could actually blow out your kidneys. Change in host microbes. This is the symbiotic changes that occurs in us. So it could be how one bacteria changes locations and it goes from being really good to really bad. Host benefits from microbes. We get protection from them. So we can prevent other bacteria from growing on us. We get vitamins. Vitamin B and K come from E. coli. And they also help prime our immune systems to keep bad things away from us. Transient flora are going to be those ones that we pick up from other areas, and we've kind of gone over this. The places that we can get it from is human reservoirs. So this could be from sexual or just being picked up from a person sneezing. Animal reservoirs, so we can get these from animals and get picked up. So like for instance Lyme disease, we get that from a deer, but the deer is bitten by a tick. That tick then comes bites us, and that's how we get Lyme disease. And then non-living reservoirs could be something like valley fever. You breathe the spores in and it gets into your lungs and grows. Tetanus, which is from soil, and it kind of comes in through a cut. Amoeba, which is water contamination. Transmittance of these comes in from different places. Direct, indirect, or droplets. Direct would be where you're touching somebody and you pick it up that way. Indirect would be from fomites touching you or something from the outside touching you. And then droplets make sense. When you sneeze, you have a lot of these viral particles fly, and they are designed to infect a new human person. Vehicles of non-living medium would be water, airborne, and foodborne. These are going to be things like somebody drinking water that's contaminated, food that wasn't prepared correctly and gives issue, and airborne. These are going to be things that are perfectly in the air, get picked up into the air, and then, so it's in the soil, sorry, gets picked up in the air and then inhaled. Other vectors are going to be animal or insect carriers. So this could be, this bug was sitting in fecal material, then stepped on your burger, you ate it, and then that's how you got a stomach issue. Biological transfer is going to be getting a blood transfer from a mosquito. Luckily, HIV doesn't come this way, but we do get malaria and a few other things this way. And it's just this mosquito bites somebody with malaria, flies off, a day or two later bites a new person, and then transmits the malaria disease.